All right, now on to part two of photosynthesis, the dark cycle. So this is what the plant cells do when there's no sunlight, how they get, sugar and energy and stuff. Um, oh yeah, slide 22, draw that diagram. 23, carbon fixation reactions. So they occur in the stroma as opposed to the chloroplast. During the Calvin cycle, energy is needed to build carbohydrates. The energy is supplied from the ATP molecules that get made during the light reactions, and also the NADPH, which is a byproduct of photosystem 1. Watch these videos here, they'll help explain it better. Calvin cycle. So carbon enters the cycle as CO2 and leaves as a sugar called G3P, like glyceraldehyde 3-phosphate, which is it's a 3-carbon compound. For the net synthesis of 1G3P, the cycle has to take place 3 times to fix 3 molecules of CO2. There's this little cycle here. Definitely draw it. Here's another one on slide 25. So 3 CO2 enter, they get fixed to make 6 times 3 carbon. They get reduced to make 1G3P. Then 5 of the 3 carbons regenerate the CO2 acceptor to make 3 5-carbon things, and the cycle continues. Slide 26 has a much more in-depth and probably better cycle. Rubisco, I'm sure Dr. Briggs went over that. There's really, um, actually, I'm gonna get to that on the next slide. So, just draw these cycles and diagrams in a way that you can understand them. All right, photorespiration with Rubisco. Um, so normal temp, which is 25 Celsius down, um, it does CO2 fixation and produces carbohydrates. At higher temperatures, with increased oxidation concentrations, has increased oxidation of RuBP and production of CO2. So its affinity for CO2 is around 80 times greater than O2, which is a lot. In most plants, C3 plants, initial carbon dioxide fixation is used through Rubisco, forms a 3-carbon compound, that's the G3P. In photorespiration, Rubisco adds oxygen instead of carbon dioxide in the Calvin cycle, which makes a 2-carbon compound. It consumes oxygen and releases CO2, and there's no ATP or sugar production. So when Rubisco does that, it's kind of annoying and dumb, and Dr. Briggs hates it, and you should too. Um, C4 plants fix carbon using an enzyme besides Rubisco. It uses PEP carboxylase. So 4-carbon compounds are produced. CO2 is released and used by Rubisco in the Calvin cycle. So it helps Rubisco not fail at its job, is what the PEP carboxylase cycle does, aka C4 photosynthesis. So here, slide 30 has a little diagram for C4 photosynthesis. Note the lack of Rubisco. Definitely draw that. All right. Cam plants. Some plants, like succulents, they're kind of, think like your desert plants that are good at being desiccation resistant. They don't dry out. They're good at conserving water. They use crissolation acid metabolism, CAM, to fix carbon. So they open their stomata at night, and that helps them incorporate CO2 into organic acids. Then during the day, when it heats up, they close their stomata and the CO2 is released from the organic acid and used in the Calvin cycle. And that's how they conserve water. Um, these last couple of slides here, starting at 32, and I'm assuming to the end, is just little diagrams on how cells divide. So, slide 33, there's like three stages. There's reproduction, growth and development, and tissue renewal. Um, and then bacterial cell division, bacteria divide by binary fission. Next week, Dr. Briggs is gonna take you into all this kind of fun stuff. So, hope these are helpful, and I'll see you on Monday.